Hello and welcome to Nikolay's genetics lessons, and today I got a question from one of my subscribers. When a DNA digests by this particular enzyme, fragments on average is 1,048,500 base pairs in length are produced. What is the most likely number of base pairs in the recognition sequence of this enzyme? Now imagine that cell has infinite supply of 4 bases: adenine, guanine, cytosine, and thymine, and all of these bases are in equal amounts. That means 25% adenine, 25% guanine, 25% cytosine, and 25% thymine. Now imagine that we have DNA strand, and for example, let's assume that we have such a restriction enzyme that cuts this DNA where it finds, for example, adenine. And we just take random place here, for example, here. And what is the probability that at this place we will find adenine? And you would say that chances would be are equal, that this is going to be adenine, guanine, cytosine, and thymine, because they happen in the same amount, in the same frequency. So probability that we will find here adenine is going to be 1/4, right? As well as guanine, for example, or cytosine and thymine. But what if recognition site for restriction enzyme would consist of 2 bases? And let's say that this restriction enzyme would cut this DNA whenever it's going to be 2 adenine in a row. And again, probability that we are going to have here adenine is also going to be 1/4, because we have infinite supplies of each base in a cell. So this is a probability problem with a replacement. So again, 1/4, and in this case we will say that this endonuclease would cut this DNA every 16 base pairs. And if, for example, we will have recognition sites that consist of 3 base pairs, and let's say this is going to be thymine here, then again we have to multiply by 1/4, and in this case we are going to get 1 every 64 base pairs, 1 cut per every 64 base pairs, with 3 bases in recognition site for restriction enzyme. So now I think you got the idea, and we can build simple table. If we have 1 base recognition site, for recognition site, then endonuclease would cut our DNA every 4 bases, with 2 every 16 bases, with 3 every 64 bases, with 4 every 256 bases, with 5 every 1024 bases. With recognition site which consists of 6 base pairs, we are going to get 1 cut every 4096 base pairs. With 7, we just have to multiply this number by 4, and this is going to be 16,384 base pairs. With 8, again, we have to multiply this number by 4, and this is going to be 65,536 base pairs. With recognition site which consists of 9 base pairs, we are going to get 1 cut every 262,144 base pairs. And with 10 base pairs, recognition site which consists of 10 base pairs, again we have to multiply this number by 4, and we are going to get 1,048,576. And as you see, this number is very close to the number in our problem, but not exactly the same, but very, very close. Why? Because this is just a statistical number. It can be a little bit less, a little bit more, but overall have to be close to this number. And as you see, the number we got is very close. But I think that you still have a question, because DNA is double-stranded. What if certain sequence also would happen on the other strand of the DNA? Should we, in this case, multiply by 2 number of recognition sites? Take a look what actually happens. Imagine that this is double-stranded DNA, and let's say we have here adenine, adenine, thymine, thymine. On the other strand, corresponding strand of the DNA, we are going to have adenine, adenine, thymine, and thymine, because adenine base pairs with thymine. So let's say we have here 5' end and 3' end here, 5' end here, and 3' end here. In most cases, such recognition sites would be palindromic. Take a look. From 5' end, here we have adenine, adenine, thymine, thymine. Here from 5' end to 3' end, we also have the same sequence, adenine, adenine, thymine, thymine. So in genetics, we call palindromic sequence not the sequence that reads back and forth the same on the same strand of the DNA, but which reads the same on other strand of the DNA, AATT, and here AATT, from 5' end to 3' end. So this is palindromic sequence, and most restriction enzymes cut in such positions where sequence is palindromic. Let's say in this place. In the case of palindromic sequence, no further mathematical calculations is needed. But if sequence is not going to be palindromic, for example, again, let me use different example. Let's say we have A, T, C, G. Then on the other strand of the DNA, we are going to have C, G, A, and T. Again, 5' end and 3' end, 5' end and 3' end. And as you see, the sequence is not palindromic, because here we have ATCG and here we have CGAT, which is not the same. So in this case, we are going to have twice as many recognition sites for such restriction endonuclease, and we have to multiply our answer by 2. But because in our problem is nothing said whether it is palindromic or non-palindromic sequence, we have to assume that this is palindromic, because most recognition sites are palindromic, especially those which are chosen on purpose for commercial applications. Plus, let's assume that our site is not palindromic, and for example, we have 9 base pairs in such a site which is not palindromic. Then we have to multiply this number by 2, and we are going to get 500,000, which is not close to this number at all. And if recognition site consists of 10 base pairs, and if it is not palindromic, then we have to multiply this number by 2, and we are going to get 2 million. So we skip this answer with non-palindromic sequence. The closest would be 500,000 and 2 million, which is not close to our answer. So we have to assume that this answer would fit ideally our problem, that we have 10 base pairs in recognition sequence for restriction endonuclease, and this recognition sequence is going to be palindromic. And this is all for today. Subscribe and see you in the next video. Goodbye.